Good afternoon, everyone, and welcome to the Cambridge Center for AI in Medicine. I'm Mihaela van der Schaar, and I'm the center director. If you are not familiar with CCAIM, we started our endeavor 1.5 years ago in January 2021. And we are a unique center in Cambridge because we span machine learning and artificial intelligence, medicine, and science. So we are a very interdisciplinary group. And we aim to really push boundaries across these three domains. Cutting edge machine learning is going to push the boundaries of medicine, and conversely, problems in medicine and bioscience are driving cutting edge research in machine learning and AI. So it is in this context that we have also this distinguished seminar series. And it is my great pleasure that this academic year, we start the seminar series with Professor Yu. Because really she is a pioneer at the frontier of these three fields. She is the Chancellor's Distinguished Professor and Class of 1936 second chair in the Department of Statistics, as well as Electrical and Computer Science, and the Center for Computational Biology at UC Berkeley. She leads the U group, which consists of 15-20 students and postdocs in statistics, as well as electrical engineering and computer science. She was formally trained as a statistician, but her research goes way beyond that, and she's pushing the boundaries on numerous other areas as well. And together with her group, her work has leveraged new computational developments to solve important scientific problems by combining novel statistical machine learning approaches with domain expertise of her many collaborators in neuroscience, in genomics, and in precision medicine. And she and her team developed relevant theory to understand random forests as well as deep learning insights from insight to practice. Bin has numerous awards. Inclusively, she is a member of the US National Academy of Science, the American Academy of Arts and Sciences. She is the past president of the Institute of Mathematical Statistics. She is a Guggenheim Fellow, a Taki Memorial Lecturer of the Bernoulli Society, Ritz Lecturer of IMS, as well as COPS L. Scott Prize winner. She holds also an honorary degree from the University of Lausanne in Switzerland, and she served on the inaugural Scientific Advisory Board here in the UK and the UK Turing Institute. And she also has been serving on the editorial board of the PNS. Bin, it is a great pleasure to have you with us today and to start our seminar series in CCAIM this year on such a high note. Thank you so much for being with us.
It's a great pleasure to visit Cambridge again, but this time virtually. So I know that this is a very interdisciplinary audience, and that's the audience I kind of usually work with these days. So do feel free to ask questions. So during the talk, I think it's much fun to have a discussion than just for me to go through my prepared slides. So today the topic is on predictability, stability, and I'll connect with causality. And I will use the background or ground the talk through a case study that our collaborative research we have been carried out with a cardiology group from Stanford to find a genetic driver of heart disease. I like this quote by Bill Gates: AI is like nuclear energy, both promising and dangerous. And precision medicine or digital health is one area that we can really make huge progress, but at the same time, human lives are on stake. So we really need to make sure that we minimize the damage. Most of my group's research for the last 1 or 2 decades been on biomedical problems. And this grounding project started 4 years ago from Charles Zuckerberg BioHub Intercampus Award with PIs across Berkeley, Stanford, and UCSF. The many people took knowledge. Let me just call out for Yu and Ashley has been the main collaborator, and also his younger colleague earlier, James Priest. And then the young people leading the effort has been Tiffany Tan, who will be on the job market this year, and also Qianru Wang, who's a postdoc from Yuan's group. So the philosophy in my group right now is really try to solve problems, and especially medical problems. And this heart condition called hypertrophic cardiomyopathy, HCM, is a specialty of Yu and Ashley lab. And what goes wrong with heart is that the left ventricle wall gets thickened, right? This is a normal heart, and this is a disease heart. And because the wall gets thickened, it's harder, much harder for the heart to pump blood. And many things happen. You can faint, you can have a heart attack more easily, you can have heart failure. So it's a condition happens in the US, 1 in 500 people. And the role of my group for myself has been giving the, I would say, the engine behind a lot of AI algorithms, which is data-driven decisions. And we combine medicine, machine learning, statistics with a lot of genomics and medical knowledge. If you take a step back to see how do we make AI safe or trustworthy, you can see that there are probably two broadly classified approaches. One is we do the best we can in the process before we write any decision rules. The other is damage control, right? Something goes wrong, we find out what happened and we fix it. So I think both approaches are necessary, but we hope we maximize the first approach so we minimize the chance that we actually have to do damage control. So what I would talk about the general principles fall into the first category. And data science or digital health, it's really a whole process. It's not just one-step modeling, which is emphasized a lot typically in research academic community. People talk about algorithm, which is hugely important, but it's not enough, right? If all the early steps are not taken care of properly, the fancy algorithm won't save us. And at the heart of all these different steps in a data science lifecycle are people, right? People make so many human judgment calls, and they create actually much more uncertainty than the traditional probabilistic or statistical machine learning framework are accounting for, right? Data cleaning, you say you have my group there, we clean the data, we're gonna end up with like 20 different versions. And that's extra uncertainty that we need to take into account. And also in visualization, we look at different plots, different colors will lead us in different paths. So all of this, we need to take into account. I would like to advocate that there are two very important common sense axioms in data science or any data-driven work, which is stability and reality check. So scientific or medical conclusions should be stable to human judgment calls. I hope nobody will argue against it. Unless you write a document saying that this is a better human judgment, and then you document it. And you need to really have an integrated data science life cycle that like these different parts who don't talk to each other. And we should really, the conclusion should really correspond or capture reality. And if we want a quality control data science life cycle, we need minimum reality check and stability check. And stability is very broadly defined, it's really principle and also reality check. So it's not, this is like, you have one metric, you can have a variety of metrics, then you find the right ones for your problem and you define these different checks. So we started using the term in our paper with Carl Kumbir, 2020, we call vertical data science. Vertical is a word which is not common used in English, but it's very much used in Spanish, means truthful. And this is really a term we try to put there for people to rally to use for quality control. So we define the paper as vertical data science extracts reliable, reproducible information from data with an enriched technical language to communicate and evaluate empirical evidence in context human decisions and domain knowledge. So it's very much a holistic approach with quantitative and qualitative arguments. And we want to realize the promise of AI data science and mitigate the dangers. So let's go back to the HCM problem, right? So we set up the goal. Actually, we already changed the goal. There's an iteration already. First, we're looking at another phenotype, and we didn't see any signal. So we changed the phenotype after we got the grant to this HCM. And of course, the choice of this problem was already driven by you and Ashley's subject knowledge, saying that there should be some genetic signal. It's a genetic related disease. It's not something hugely impacted by environments, but we could be wrong, right? So we decided to use the UK Biobank data, and much discussion and a lot of early preliminary work. And we set up a protocol to work with Ashley lab and other scientific labs we call with negative controls. And we also use an annotated database. What do I mean by negative control when we interact with Ashley? Informal conclusion sharing. We don't just give them what we found. We give them three, say, lists of genes. One is what a models or whatever we decide to recommend, and usually it's the rank list. We take also the same size from the middle of the ranked list, and then random selected group. So we give them things that to see whether they can really tell this three lists apart. And so far, my collaborators haven't failed. So not just giving them something with a model find to avoid confirmation bias. And then we do knockdown experiments in Ewan's lab. Sorry, somehow the numbers messed up. So for the rest of the talk, I will talk about introduce the PCS framework. You might have heard early versions of our work because I've been talking about this PCS framework for a couple of years now. So this is a more updated version. And the PCS framework for predictability, computability, and stability, I'll get to causality later. It's really a general framework at the level of principles. It's like you do maximum likelihood, you don't have particular formulas, but it's a principle guide you for a particular problem. So it's a similar approach. And you can use it for new methodology development. I like the in-context approach. That means you solve a problem and then develop general methodology, solve this particular problem and can be generalizable to other problems. And then I go back to the HCM problem to see how we find recommendations, recommended genes, and also report on experimental results. So finishing a paper we plan to submit to Nature Medicine on this work. And now I'll end with some software we developed for to make PCS analysis easy. So as I said, PCS means predictability, computability. It's really try to combine good ideas from machine learning and statistics and then expand significantly on both in terms of uncertainty assessment. And also reinforce what we do with cross-validation for model validation, model check, or relative check beyond that. So the paper came out in 2020. The paper took like 3 or 4 years to write. And it's really used predictability as at least the first approach to reality check. You should really argument it with domain knowledge, downstream analysis, all the other things. So predictability is kind of a minimum requirement for reality check, but it's not the only thing. And computability, right? We need to compute. And we also add compute data-inspired simulations, not just for convergence and speed and scalability. Go beyond CS so that you can simulate models that you know the truth, use data, and then see how your methods work. And stability is a significant extension of the ideas in statistics, sample-to-sample variability, robust statistics, model-model variability, and also from control theory. And it's a minimum requirement for interpretation and also causality. So we try to unify machine learning and statistics and expand on both. And documentation is a huge important component of the framework because this is not a cookie cutter approach. It's really help you with principle to think in the context of your problem. So you have models, they are mental construct until you establish the connection with reality through both quantitative and qualitative narratives like R Markdown or Jupyter Notebook. So you really make an argument. Actually, these models are reasonable because that's related to my domain knowledge and not the other kind of models. PCS connects science engineering because predictability and stability embed scientific principle with prediction replication. As I mentioned earlier, predictability is definitely practically necessary and also include data-inspired simulation. That's why we have a software package called SimShelp to make simulation in traditional or more standard model simulation easy. So I've been working using this framework with many collaborators. We have like 8 or 10 projects using this principle, but you have to do a lot more work to really make it work. And have been collaborating pretty closely with Adrian Kompleth. It's an ER doctor from UCSF. So he kind of saw me out after hearing a 10-minute talk at the UC Medicine, AI Medicine about 3 or 4 years ago. So I asked him once, like, why did you reach out to me and why you keep working with us? That's what he said. I think it's kind of really says from the user point of view why he liked the framework. He said the PCS framework builds a working relation between data and the clinical world. PCS is a look under the hood to ensure that the conclusions find out what the data genuinely suggests. In all, PCS is a holistic approach to helping the clinician understand, interpret, and build the science we need to help our patients. And stability principle on top of predictability, which is already hard of machine learning, is really a generalization of reproducibility from coding reproducibility and method generalize like reproducibility to the whole pipeline, to problem formulation stability, right? Linguist stability, when two teams work together, matrix for cancer scientists, it means some biological structure. For us, mathematical trained people, it means a tabular data set. So all of that language level is already for two teams to work together. You need to have that stability of language. Like ML, when I was a graduate student, means maximum likelihood. Right now, even I talk about ML using machine learning. So language evolve and the team work together have to understand the English or other language words in the same way. Otherwise, you can have miscommunication. And every step of the way, it's a common sense principle. And we should entertain a lot more perturbation to data and to models than we currently do. And robust statistics, people often ask me, how does it relate to robust statistics? So PCS really extends robust statistics. Robust statistics is kind of looking at a specific form of data perturbation. And if it's relevant, that's what you use. So you can go back to robust statistics. But we consider the whole pipeline of so many qualitative and quantitative perturbation from problem formulation, data cleaning, EDA. You use two different set plots for the same data with different colors, you get different impression. So I'll show you my class with the Hurricane Ian just landed in Florida. Washington Post and New York Times both have this surge, kind of storm surge map. And Washington Post used blue color. It didn't really cause the same reaction to me than the New York Times used red color. You just feel like this is very serious. But blue color, kind of nice, you get a different impression and you might do different things. So all of that needs to be taken into account, even the exploration stage, and documented. And if I really want a highbrow quote, you can go back to Plato. In the manual, he said, knowledge needs to be tied down. That's different from correct opinion. So tie down, I mean stable. You cannot talk about knowledge which is translate changing second to second. But of course, scientific knowledge does change over years and decades, but not in a very fast way, like every minute the scientific knowledge is changing, then we won't call that knowledge. And from theoretical point of view, stability is also at the heart of theory. Central limit theorem is actually a stability result, right? There's this very nice proof, Lindenberg swapping trick. You can swap variables in and out, and then you get a Gaussian swapping in, then you basically have central limit theorem. Concentration inequality have stability condition, random matrices, right? The proof's very much stability driven, and you have uniform stability for generality. So this is not just empirical common sense, it's also mathematically how we laid our foundation. And stability is a conceptual principle. It's not like give you a stability measure. For all the projects I've worked on, we use different metrics to measure stability, and we define different relevant perturbations. So a lot is about you define a reasonable perturbation. You said the data could have been collected this way. Bootstrap, I would say it's a special case of stability-driven analysis, stability analysis, because you argue under ID conditions, your bootstrap sample is as if they are from original data. But if you look at data cleaning, then you have to go upstream, right? That bootstrap doesn't cover the uncertainty introduced by two people clean the data, have two different versions of the data, and you define the metric. So it's very much help you to think about these issues, but you have to go back to your context and argue, write a document about why this is the right metric, or maybe multiple metrics. For COVID prediction problem, at the county level, we did 2 years ago, actually use different 3 metrics. We want to make sure our prediction model works for both the absolute death count difference and the relative death count difference, and also square root, because our model is used a square root transform first. And connecting to causal inference, supervised is actually a stability condition. You're saying that there's one version of the data and depends on other people get the drug or not, doesn't change your outcome. And invariance goes back to in the 40s econometrics. It's also a stability condition, also used in genetics, right? You want to find gene network that's invariant across different species, or you want to find some good predictors under different economic environments, right? So this a stability consideration. And why do we do randomization? We actually create stability or balancing of covariates. And propensity score just use context to assume conditional stability. When you assume condition on certain covariates, and you assume that you have a random toss depending on the covariance. And in causal inference, people already do sensitivity analysis, right? So all of this are saying that this is PCI try to unify all these different considerations and make them more in the same language, and so we can compare. If you take a deeper look at causality, right, probably the best causal model is Hooke's law, right, from physics, right? You put a load at the end of the spring, and we can predict how much the spring will enlarge itself. But it doesn't work every time, right? You get unlucky. The spring is old. You put a load, it breaks. So for that incident, actually, this law doesn't work. So all you need condition to make sure your spring is good shape and all of that. But still, it's very reproducible. And then you look at the other extreme of statistical causality. FDA dictates that we look at the average effect at the group level, even though we're now going to heterogeneous treatment effects, all of that. But we're getting into closer, but there's a whole spectrum. So for me, I like the definition causality by Nancy Cartwright, maybe earlier than her too. Basically saying that causality is you have enough evidence to act. So that makes it very practical, very pragmatic. It's not like you always have to find proved like mechanistic reasons, like smoking and lung cancer. We still don't know the mechanistic model for why nicotine or something else in tobacco causes cancer, but we act. We put labels on cigarettes, we tell people not to smoke. So that's in that sense, causality. NIH committee in early 60s actually made a decision, look at 23 or 25 observational studies and decided smoking caused lung cancer. So that's just enough evidence to act. And we know that predictability is not for causality, right? There are many things, you have confounding factors and all of that. However, if you add stability on top of predictability, I think we're a step closer to causality. And that's what we have been using to generate interval hypothesis or become recommendation system for experiment. So we do appropriate stability analysis on top of predictability, which is reality check. And that's how we're going to go back to HCM. Right, so this is different from conventional causal inference. So we have observational data, right? UK Biobank data is not randomized trials, it's observational data. So we use that and stability and then we do knockdown experiment, and we establish causality. But if you look at conventional causal inference, people actually use context much more qualitative and make independent type of assumptions and then draw conclusions. So this is kind of complementary approach, because not all the time you can do knockdown experiments or follow-up experiments. So our approach can also be used for clinical trials or even for further observation study because of recognition system. And I just feel more comfortable with something harder like knockdown experiments, but definitely you can do great work as well to the conventional causal inference. But I hope there's some downstream validation instead of you rely on the assumption and you estimate the effect size. And we also push PCS to causal inference randomized trials. And we actually have something we call statistics paper. So the challenge there is to define P for reality check, because in randomized clinical trials, you don't have both treatment and control outcome observed. And we actually look at a very extensive stability study over 17 different heterogeneous effect models. And then we also use external study to validate what we find. So this was led by two fantastic young people, David Vadi, now at postdoc Harvard and MIT, and he'll be on the market. And Yan Shu Tang just started a tenure track job at National University of Singapore. And this is co-led by my colleague, David Madison. So back to the more general case of data science life cycle, right? So you have all these different steps. Every step you worry about stability, and you also worry about reality check. So for problem formulation, you want to think about what's the future reality. Your digital health instrument will be useful. Always keep future in mind. So P is relevant even for problem formulation. It's not just about data in hand, it's about the future. And you want to set aside your data as close as possible as proxy to the future, not just always do random split. Often you do group split. Different hospitals should become the test set, because in the future, your clinical decision will be used for a different hospital. So all of that is P, the reality check you want to keep thinking. When you do data visualization, you want to do two different runs, some data split, and see whether the same pattern reappear. If not, then you're looking at noise. So this is consistently looking at the two angles, reality check and stability. And you try to minimize the impact of human judgment calls. Eventually, there is a meta human judgment call on many of that, but you want to remove some trivial ones. I cannot hear you.
Sorry,
is there a question?
We move on. Yes, we got one question
yes.
from Jason about the causality definition, which he thinks from Nancy sounds special. What's your opinion on different schools of causality? Pearl Rubin, discovery inference, potential outcome frameworks, structural equation models, etc.
I'm very much like a plural person. I usually want to look at, I'm not philosophically married to any approach. I want to see that if you believe what this approach, show me the empirical evidence, and I hope for the same data, same problem, based on the same domain knowledge, different approaches, sometimes reach the same conclusion. And when they don't, I think we need to go back to the drawing table and discuss what assumption you actually made in your approach that I'm not making. And then let's compare notes and then have a debate about which one is more reasonable and resolve the differences that way. Or just leave like these factors we just don't know. These factors we think are genetic drivers. We believe because both approaches agree. And the other ones we just leave as future for future exploration. So I'm very empirical in the sense that I'm open to different approaches. And there's some approaches I'm more comfortable with, become more experienced with, but don't kind of exclude other approaches. So for causal discovery, right? So I have been staying out just because I want to see more evidence, like with these graphical models. So if you ask me right now, I'm worried about that, but I can be convinced that why it works well. I hope I answered the question.
Thank you very much.
Just really what empirical evidence you can show downstream. We can keep multiple lines going and then compare notes. And it would be best to compare for the same problem, take different approach, and we can really compare. I also through simulation, but simulation is not enough. I need a real context to compare the different approaches. Okay, so this is an example, very famous example by two Harvard economists, wrote a paper 20 years ago, no, 10 years ago, for the last financial kind of meltdown. And they basically look at country year data. They had a conclusion, the high debt GPT ratio is bad for growth. They look at very high debt ratio, 90%, and then reach that conclusion. 4 years later, a graduate student heard it and his advisors tried to replicate and couldn't replicate. And this policy was used to support austerity policy in UK and Europe. And the problem was they somehow just made 3 data points from New Zealand. And there's some coding errors and conventional weighting together, but the 3 data points very important. But even I look at the original paper, I wouldn't have reached the same conclusion actually. I think they were kind of confirmation bias there. If you use the median, look at the high debt ratio, then you don't get the same conclusion. They use the mean, so a few data points really put the conclusion. So even with the data they had, I would have reached a different conclusion, or not clear would support their conclusion. But missing data points was a problem. And you look at my problem later, we're going to count cells because we do knockdown experiments. The conjecture said the genes, the reason the left ventricle wall become large is because the cell sizes become large. And then you have high resolution and low resolution, very different, right? So you have lots of data perturbation might change your conclusions. And as I mentioned, right, you can do random splits, very popular machine learning, but I think we need to move away when necessary to other split, group split for different hospitals and time split. So the key is you want your test data to be the best proxy as possible for your future data. Always keep future in mind through I'll call prediction and P, right? And you might want to do multiple ways, not just one way, because different researchers might do differently. You want to cover that uncertainty or study that. And for this problem statistics I mentioned, we did a particular way. We did actually both random splits, and we also did a group splits, and it got the same conclusion. But it's a matter of judgment call, right? Why I made into 5 folds, not 10 folds. So it depends on how many data points we have, but you always still have a judgment call. We cannot completely remove it. And different people use different methods, right? And climate scientists, I'll call, already doing PCS inference. They have 9 climate models for global temperature rise from 1.5 degrees to 5.5 degrees. And this is a lot more informative than just using one model. This is very reputable 9 groups, right? So with the software available, so one team can now also do, maybe not for this problem, for other statistical machine learning problem, can really run different methods. Instead of, oh, that's the method I like and I use, therefore I'm only going to show that. People often ask me, right, if you do all these perturbation paths, you're going to have exponential explosion, which is true. But it's not a reason not to do it. At least you do 2 paths. Instead of, oh, there's exponential explosion, therefore I don't do any. So actually we can do maybe 10 paths, right? Every step you have multiple forking. And then you either say this fork direction is not reasonable, you write a document about it. Or you just do some sample. It's a bit like Bayesian now. You sample, if all the paths are equally good, and you sample some. At least I think you should do 2 paths. At least you have 2 versions of clean data. I have my class, 3 teams clean the same digital data from some traumatic brain injury. And 3 teams end up with 3 different data sets. One team cleaned away 23% of data and got the best result on their clean data. But I don't think that's really usable, right? You clean away too much data when you use it to other real hospitals. That's not applicable anymore. You cherry picked somehow clean data. So I'm really advocating for 2 clean data sets, which we're still in the process struggle implemented, but in my class, definitely, we have now 20 students, each clean the data. We're going to continue with the same data set for the rest of the semester, and we're going to compare the conclusion. We have 20 versions, and you're going to think very different. And there's also a paper, like 29 teams try to clean this to work on the same data, get all kinds of conclusions. So this is extra uncertainty. And if you can remove that, I think the result will be a lot more reproducible, and you do documentation. So back to the HCM, right? We first wanted to use the HCM label, right, which is in the biobank. And we spent like 6 months trying to find signal. We end up with 50-50 random toss in a balanced case. And it's because many people actually have HCM were not diagnosed. We think that was the reason. So we went back to the drawing board, reformulated the problem, and Weston in Euren's group went to the MRI heart images and extracted the continuous phenotype, which is the volume of the left ventricle chamber, the wall, right? And then it's a regression problem. So we did our prediction, and there was we don't know whether we got a good model because we don't know what's the noise ceiling. We don't have replicates. So then we came out with the idea that we should binarize to a simpler binary problem. High volume, it's called left ventricle mass, low left ventricle. We did normalize for weight and height because otherwise it's not comparable. And then we create a balanced binary classification problem. And then we know 50% is a benchmark. And we barely beat it. We have consistent 55%. So this was the lowest prediction score I have ever seen in my real data analysis. I felt not great, but it's a very hard problem. So it's possible we couldn't get anything better with all the other factors. So we tried lots of stability analysis. We saw a consistent signal of 55%. So, and we look at annotated database, look at the genes, and we decided to do experiments. So that was pretty not high, but I was surprised that we actually got good signals, at least in this case. So Tiffany really led this effort, right? Reformulated the problem, and we tried top 20%, top 15, top 25, and we got similar result, right? So we want to make sure this is not the artifact of our cut threshold. So here's the UK data. You guys probably familiar with SNPs, and that's 10 million. So we have to do some dimensionality reduction. We end up using the more typical GWAS to reduce. And after we use GWAS to reduce to about 1,000, we then use a non-linear model selection, which actually our contribution to the Intercampus Award and led by the paper came a couple of years ago. But soon now, Assistant Professor Cornell Carr now at UCS postdoc, my longtime collaborator Ben Brown, which is the co-lead of the project. So we try to do pattern discovery, not like pattern recognition in computer vision, because we don't know what it looks like. And we know in genetics, in Drosophila, that's in the paper, that you have 4 order interactions. And there's also something from biology called French flag model, saying that thresholding is an important process for different biomolecules to interact. And mathematical match to that is decision rules, or decision, like a Boolean type of interaction. So that's why we went for the random forest. And more we have lots of different nonlinear methods, give very similar prediction performance. So we have to match now at the more detailed level, the building block of a process to the domain knowledge. That's where I think we're going to have a lot to gain instead of just let's look at prediction error, because we really want to have follow-up experiments. We want interpretation, we want mechanistic explanations. So all of that, we have to go to the next level, not just the prediction accuracy. And random forest, everybody knows, so I won't say much. So what we did, people already using this idea that 2 genes fell on the same path, we consider them as interacting. But they couldn't make it interpretable because very, very unstable, even though random forest give very good performance for genomic problems. So we added stability. So this was actually one of the motivating examples, case studies for us to arrive at the PCS framework. We're working on with the neuroscience problem, this problem, to see whether these ideas actually work before we wrote a paper about PCS. So we use self-dimensionality reduction, use the MDI, the importance index, instead of uniform sample from all the predictors, we sample based on the weight. And then we have a cool random intersection tree actually from Raj and Schaar, from Cambridge and Mannhausen, from ETH to find interactions. And then we have an outer loop of backing to assess stability. So all of that look at the result for Drosophila. So the left, you see the RC curve. So you start with random forest, and then you downweigh the unimportant ones, but you still keep them in the loop. And then you upweigh the important ones. And we did the 3 iterations. So you see the 3 curve is on top of each other. We didn't lose any prediction accuracy. And if you look at the right, which is a stability score. So the red ones are the pairwise interactions, and the blue ones are 3-way interactions. So we find plenty. We found 20 pairwise interactions. And luckily, before we even collect the data, in the Drosophila literature, people already prove use experiments. 80% of them, or 16 of them, already proved by wet lab experiments. It's before we even collected the data. So this is a good success. And 4 of them, I think 2 are expert. Sue Seneca think that probably were right, but we don't know. The 2 probably we got it wrong. And the blue ones become recommendations, right? People are just beginning to do 3-way interaction experiments. And we put in all our results for enhancer. That's for enhancer problem. And on the UC Santa Cruz genome browser track, we're finishing this paper, which has been on archive for 4 years. So other Drosophila scientists could use to say, oh, this is recommended. And we have high peaks. And maybe I want to do some experiment based on my knowledge. So this is really put it there to be used. And we also done some really nice piece of theory. Actually, just, sorry, it should be updated. Just appeared in PNS that we have a theoretical version of iterative random forest. We show that if you have a Boolean model with linear combination Boolean function, we call LSS. It's a new model study random forest-like things. Then we can get the interaction without ever estimating explicitly the continuous parameters. It's kind of really amazing that we could do that. And it's really work on my former postdoc, who is the professor back in Germany, and Hugh Wang and Xin Li both went to finance, one to Sigma, one to Volin. So back to HCM, right? So as I said, we did GWAS, bought LMM, reduced it to 1,000, and then we changed the problem into binary classification from continuous phenotype and use iterative random forest to extract candidate gene interactions. And then we developed some new stability scores too in the process, not in the RF. So that's the genes we find. And Tiffany and Abby and Shell were really leading the efforts. So we find genes, one is T10, which is a well-known heart muscle gene. And growth factor, kind of known, kind of not known. And then we find promising candidate CCDC141, which is next to T10. But we discovered, people thought they have the same functionality as T10. Actually, there's an interaction. They don't work the same. And then we look at the databases, working with Chad Weldy from Stanford, and a lot of discussions, talked to Chad, and they pass our test. We give them 3 lists, and then we decided to do experiments. And this is the database. Find these genes, you only can find like marginal functionalities. They're all kind of related to what we were looking for. So we settled to take the 3 top genes and do some interactions, experiments, knockdown experiment. That's where Chen Ru comes in with the help of Nate, undergraduate in Yuan's lab, prepare the sample. So you silence the genes. So you have these stem cells that don't look like real muscle cells, but that's at least real on Petri dish. And then you silence genes, use this called sRNA transfection. And then you don't do CRISPR because this is cheaper. And then you see what happens. And there's a lot of work. Her work was really designed. She was a mechanical engineer from MIT. This cell sorter because we predict that the cell size is affected by this disease genes. And you can see that. Oops, sorry. I'm sorry, yeah. I don't know, it doesn't go in. You can see a little bit. They kind of go pretty fast. You have to do automation. You cannot humanly count it, right? And where did that go? Sorry, I have to go back to. Yeah, sorry about that. Somehow it should have worked, but okay. So back to track. Yeah. So she had to develop a automatic like cell finder and then find the area, the size. And we first thought that, you know, maybe we could improve this step. So we did a lot of manual annotation, with now Almer joining with Anna, who's a postdoc, also getting on the market. And we find that her actually software works really well. And then we find a problem. I hope this works. I'm sorry, but this is really bad. So one thing we discovered they didn't know is that there's this humongous cell got counted so many times because it moves so slowly. And that's what they didn't know. Otherwise, we're going to really have result that saying that the cell size is so huge, but actually they are just the experimental problems, right? So that's a big discovery. And by just going our called old-fashioned statistical work, shoelace work, my former colleague David Freeman would call, that it just goes through lots of movies and humanly look at them and make sure that we're not missing anything. They were actually a little bit taken back when I said that I want to look at the data, do the analysis fun, because they already have a pipeline. But in the end, they were very happy because they didn't have time to look at all the images. And so that's the result. So now you see that we have two lines, disease line, and the other line, which is just healthy line. And we knocked down CCDC and the Grease factor, both of them, we knocked down them. And you can see that there's a whole shift of the cell size, not just at the big size, because we don't want to see, if you use any test with so many cells, you're going to have significant, but significant now doesn't make as much sense because anything will look significant here. So it's really use the QQ plus, see a whole shift that even the small cell, the size got bigger for the squam. So when you knock down, that means it's helpful. So that means these other genes are probably positively responsive for the problem. And we have other results. And for 6 experiments, 3 turned out to be really, really obvious that we find something, and the other 3 kind of on the borderline. So we're now finishing the paper. So to finish that, we have 2 software to make PCS data analysis easy, built on Berkeley CS, like ML Flow and also Array. And then we also have a simulation package in R, the first in Python, the second in R. So these are the people develop the vertical flow. Actually, should be, now it's finished, now just ongoing. And then SimChef, this is the team who really try to make, you can easily just do data generation much more easily than you have to write a lot of your own code, but you can just click and then do different models and use your real data and then really do the analysis. So hope you find them useful. This is in R, the vFlow for the PCS analysis is in Python, compatible with many platforms. So I'm finishing a book with my former student, current postdoc, Rebecca Butter, will be with MIT Press. We definitely want to submit the version in December, and we'll have a free online copy when the book goes to print, probably in 2023. And the papers are on my website, and all the funding agencies have been supporting us. And I just want to leave you with this slide, that the PCI framework is really used with documentation to fully account uncertainty in data science lifecycle with reality check. And it's a lot more than modeling. It's not just data perturbation, model perturbation. It's also about data cleaning. I think data cleaning is actually the big elephant in the room of uncertainty that we have been ignoring. And good, I think all the good groups actually do different versions or something, but we don't have the habit reporting in paper. So I kind of want to bring data cleaning to the upfront so that we can all address it, because it's huge uncertainty when you look at how people clean the data. That's just really under-addressed. Thank you very much.
Thank you very much. Been a big applause from all of us, but unfortunately a virtual one. Thank you very, very much for the wonderful presentation. Chinyuan, I see that you have a question. Would you like to unmute yourself and ask the question yourself?
Oh, yes, certainly. So my question is, do you or how do you take into account the study design and how the data are collected in generating your perturbed path and assessing stability?
So that's where when you create different perturbation, you need to take that into account, right? The best, I would say, that be there, be the data collector, be the detective. So try to understand the process, go to the lab, see how the data is collected, and use that information to build into your data perturbation.
Okay.
Right. Of course, you make human judgment call there too. But being there will give you much richer information to know what are the crucial factors you might want to take into account in your data perturbation.
Sure, yeah.
And then you best have a document about data collection process, and then go back there when you propose your data perturbation to say, this is the stage, right? You say that actually 2 people collect this data, I'm putting them together. And that's something I want to reproduce while I do my data perturbation. I basically make one perturbation, one person, and make another perturbation for another person and put them together because there's extra uncertainty. Okay. So it's a huge amount of work, but at the same time, I think we're dealing with very complex problems, and slowing down, especially for medicine, I think would really benefit in the long run. We usually write a paper on this for like 3 or 4 years, take us, because you want it to be backed up and to be useful and can test, kind of pass the test of time. I don't see any other easy way to do it. It's not for paper publication.
Sure.
If you want to make an impact and have patients helped. For me, I would say that I can sleep at night when I do that.
If I may ask one more question about
sure.
Data cleaning on your last slide, do you see any possibility to develop like a mathematical or statistical theory to guide us in that step, or do we have to just to sort of follow some heuristic principles at this point?
Well, I think the thing is I wouldn't develop the theory for the sake of theory, but if the theory helps, I'm open to it. So I think we're too early on we now just try to, I was just proposing do 2 versions, right? So we have a whole chapter in the book on data cleaning, give you some guidelines about, you need to worry about validity, you need to worry about missingness, but then we already have a problem right there, that multiple ways impute data, and I worry about that stage. So I think I will take an aggressive approach. So if we have a software to help people record all the approaches, and you can have a discrete mathematical theory maybe with it, but I don't see a continuous kind of mathematical theory coming out of it, because people make discrete decisions. And the questions, what's the purpose of developing a theory? If the theorem will bring some insight, I'll go for it, but not otherwise.
Sure, I was just wondering if, I mean, I don't want to develop theory for the sake of it as well. It's just, I'd like to sometimes have some theory to guide my thinking.
But if the theory is not grounded on reality, it's not going to help you, right? So I think we're at the stage actually having more empirical experience.
Sure. Yeah.
I'm not saying that in the future, I'm open to it, but I think right now it's just, we don't even deal with it. So I usually like to do in context development so that I do have more experience, have some software and see what happens.
Okay. Thank you very much.
Thank you for the questions.
Anybody else?
There's somebody, is there somebody? There's a chat.
No, it seems that these were the questions. Thank you very much. I want to ask you a final question. Since you are working with so many clinicians, and this is what we are trying to do in the center as well, and you are reaching out across different departments in Berkeley. Do you have any word for advice for us? What did you find as being successful collaboration, and how to find successful collaborators in these interdisciplinary settings?
So the collaborators work well with me. The people, of course, I want to find the best ones, right? And because otherwise you're building on their domain knowledge. If they have not on top of their field, then I think it's hard for you to build on it. So, but the other thing is I want them to be open-minded. I don't want to work as a consultant. I want to work as a partner. So, I mean, Chuki said statistician play in everybody's backyard, but I said I want to play in everybody's front yard with them. I'm not playing by myself. So it's very important to have people who are open to even, my collaborate in the end will take my ideas, scientific ideas. I don't stay just with the statistics proper. I'm very much integrated, right? So UN's lab, my team work with the team as an integrated team instead of a separate team. So we make scientific suggestions. They might not like it and they give us a reason, and that's fine. But it's not like we keep away saying that, oh, that's your domain, you know everything, and therefore we stay out. I don't stay out because I'm just very curious. I want to know as much as I can. And that's why the experiment was a success story. In the beginning, they were taken aback a bit, right? We want to look at their analysis. But after we discovered these big, huge cells, we actually just removed them, right, over certain, because that will bias the result, not in our favor, so it's conservative. But in the end, they were very happy. So you have to establish credit, right? Working with scientists, my first serious collaboration, I worked with JPL people, but they were a lot more open because there was already a statistician on the team. Yes or no? But with neuroscience, I spent a whole year just doing free consulting with the neuroscience lab, and to kind of establish my credibility. I was on actually the Guggenheim, and I just spent every day, half a day at their lab, make friends with their students, with their postdocs, because the PI too busy to talk to me. So it's really, you have to pay the dues. And then they trust you. You have to build a trust as humans. First, the experimental scientists worry that you just scoop them, right? And you take their data, and then that's what you don't want. And they want to see you just curious about the science, and just you want to do your modeling without thinking. So I always want to become a science or doctor. I just always want to, the things I don't know, I find most exciting. So it's very not hard for me because I'm just very curious to learn the new things. And not hurry, just really, I say I can sleep at night. So we don't talk about paper writing very much until we get good results. And then we talk about how to communicate. I think today's research, too much, we talk about paper writing too soon. And then somehow we convey implicit idea that it's all about paper. It's not about understanding or doing something. I think the terminology does impact, language impact our mind. So it's better to talk about understanding and research and help patients. So why give this lecture? Sometimes I ask people to think, if you or your relative will be treated by the clinical decision rules, what would you do? Kind of put them in the right mindset. Would you going to do this so quickly, or you're going to be really worried about every step of the way? If you are the patient, are you going to only concentrate on the algorithm bit, not about how the data came about? I think people will naturally want to know more, the whole process. It's when you're deprived the context. Yeah. So how people put in context.
We are so much on board with your message, Bin. Thank you so very much for your lecture and for all the wise words. And we are very much following your view as well. So look forward to having you visit us sometime in the future in person and interacting with us all. Thank you very much.
Thank you for having me. Yeah.
Thank you.